I'm Catherine Roughton, a second-year master's student working in Dr. Kircher's lab, and today I'm presenting on the effects of microRNA 888 and 891A on transdifferentiation of LNCaP cells into neuroendocrine-like cells, or NE cells. To start, the main focus of our lab is prostate cancer, or PCA. Prostate cancer is a prevalent disease that is the leading cause of cancer in men and the second leading cause of cancer-related deaths. Typical treatment of PCA involved inhibiting the availability of androgen to the cancer cells, as prostate cancer cells typically use androgen to modulate its growth. Inhibition of androgen initially decreases tumor severity in patients, but the cancer will eventually develop resistance to the drug and differentiate into a more aggressive form called castration-resistant prostate cancer, or CRPC. CRPC is known for having morphology resembling neuroendocrine cells, with long spindly processes and even expression of known neuroendocrine traits. For our lab, our targets were microRNA 888 and microRNA 891A, both of which are part of the miR888 cluster. First and foremost, microRNAs are small non-coding RNAs, typically about 20 nucleotides long, whose role is in gene regulation. The typical mode of action is that they bind to messenger RNA and inhibit the translation of RNA into protein. The miR888 cluster is known for targeting various tumor suppressors and inhibiting their function. Our lab previously found that miR888 was elevated in prostatic secretions taken from the urine samples of PCA patients. Additionally, several in vitro assays showed that overexpression of miR888 causes increased cell growth, migration, and invasion. It also caused increased tumor load in mice in vivo. Because the miR888 cluster has a role in aggressive prostate cancer, my project aims to determine if CRISPR deletions of miR888 or 891A in LNCaP cells would slow NE transdifferentiation. To test this, we use LNCaP cells, which are human prostate cells derived from a lymph node metastasis. The knockout lines of miR888 or miR891A were generated, and these, along with regular LNCaPs, were plated to confluency. The cells were all plated in androgen strip media and maintained for up to 8 weeks. Each week, cells were imaged, then harvested to test the expression of the neuroendocrine markers. We found that the differentiation of neuroendocrine cells was slower as compared to regular LNCaP cells. This is indicated by the green circles in the figure, in which the top circle shows LNCaPs developing the spindly processes like an NE cell around week 1. For the knockout line 888, these processes weren't seen until at least week 2, and for knockout 891A, they were seen even later. We also found the expression of NE markers was generally decreased in the knockout lines as compared to regular LNCaPs. On the right, we see the resulting graphs from qPCR. These indicate the levels of neuroendocrine marker expression from weeks 0 to 5 compared against the expression level of LNCaP cells. The blue bars show the expression of the NE marker neurotensin. The gray bars indicate secretagoguin. Both knockouts had a relatively stable expression of neurotensin throughout the time course. For secretagoguin, however, it is vastly different from the LNCaP line. On the top graph, week 0 indicates both the knockout and LNCaPs had the same level of expression of SCGN. However, during week 1, when the imaging showed the LNCaPs differentiated, the expression of SCGN in LNCaP cells greatly increased. The miR888 knockout line did not differentiate at this time, however, which is why we see a great decrease in the expression following week 1. The same is also true of the bottom graph with the knockout line miR891A, indicating the knockouts have a much slower differentiation rate than LNCaPs. Our previous work done by student Megan Sage shows the overexpression of 888 or 891A increases differentiation rate. The red box shows both lentiviral lines having spindly processes by week 1, a stark contrast from the knockout lines. The graphs along the right show the lentiviral lines have an increased expression of the NE markers as compared to the LNCaPs, the opposite of our knockout project. Thus, this project indicated that deleting microRNA 888 and 891A slows differentiation of LNCaP cells into neuroendocrine-like cells. Previous studies have shown that overexpression of miR888 accelerates transdifferentiation. Thus, the miR888 cluster must play some role in the transdifferentiation into NE cells. To verify this, a Western blot should verify the tested NE markers. We could also stain the spindly processes to quantify their growth over time. Overall, one can conclude the miR888 cluster plays some role in differentiation, and this mechanic may provide miR888 as an ideal candidate for the anti-miR therapies against castration-resistant prostate cancer. And with that, I would like to thank Dr. Aurora Kircher for her help and guidance on this project. I'd also like to thank her past students Megan Sage and Aruba Ayaz for their work on the lentiviral assays. Thank you.